researcher at ASU, and the thermodynamic study wasn't really gone into until September 2011, because conditions at that time only allowed DNA to assemble itself in structures in 12 hours, and that was very inefficient. So instead of 12 hours, Shojing and Professor Halian wanted to be able to make these structures self-assemble in under 10 minutes with certain thermodynamic conditions. And that's why they studied thermodynamics, to make it more efficient. So yeah, it was inefficient, they sought to make it efficient. And they also wanted to learn about the properties of DNA related to energy, which is what thermodynamics is, under temperature. So this is one theoretical use of DNA, the DNA self-assembly process. So DNA nanotechnology, the design and manufacturing of organic material DNA for technological use. So one interesting application is nanopillars, and these are little DNA pillars that can be replaced in solar cells as an alternative to silicon solar cells, which greatly reduces the cost of solar energy. So that's one cool application of this. And they can also be used as semiconductors for the next wave of technology. So like display technology, so like your LED screen in future generations might include DNA in it. Like it's just like you can use DNA in technology and not just for like what its main purposes is the fundamental basis for all biological organisms. And nanomedicine, this is actually really cool. So DNA nanoparticles, they have a high surface area to volume ratio, and so functional groups can attach onto these structures and locate tumor cells for the body and mark them, so the body can have an easier time to kill these tumor cells. So one famous example is Abraxane. So it's an FDA-approved DNA nanoparticle used to treat breast cancer. So definitely there is some practical and useful applications to this, but not everything has been realized just yet. So this is the company that Biodesign works with, IDT, and it's a company centered in Iowa, and the strands are fairly expensive. Just to make, it's about $7 per strand, and some strands go per base pair, and some strands go as long as 80 base pairs, and we use about 10 strands per day. So there's like hundreds and hundreds of dollars going into this. And yeah, you can purchase strands of 2 to 80 base pairs long. And thermodynamics uses generally larger structures, so all of our base pairs were around 60 to 80 base pairs long, all of our strands. So it was fairly expensive. But this is just the company and a little bit about it. And this is the molecule I work with, obviously, deoxyribonucleic acid, DNA. And the part of the DNA molecule that makes it such an efficient structure to use for technology is the nitrogenous bases, adenine, guanine, cytosine, and thymine. These 4 bases are very flexible, so they can be structured into architectures that the researchers want based on the conditions that they set for. So what type of DNA do we use? So we use DNA oligonucleotides for the self-assembly process, and they're just short RNA or DNA particles used to study the structure and properties of DNA. So, and they're created through the use of solid-phase chemical synthesis. I am actually not sure how that works because they do that at IDT, they don't actually do that at Biodesign itself. So, but yeah, the process is very complicated, so a lot of stuff going into this. And overall, it's a bottom-up process. We're not taking large structures and breaking them down and studying them. We're taking oligonucleotides, which are very simple strands and structures, and then we're using multiple strands to create and self-assemble into DNA architectures. Okay, so the first day I came to Biodesign, my researcher was using these oligonucleotides to make self-assemble a smiley face. And it had no practical use, and the smiley face costed $1,200 to make. It had no practical use other than just to make a smiley face. So that's another thing about research. A lot of funding goes into it. I was very surprised. Because if you can make a $1,200 smiley face for no reason, that's pretty cool stuff. And yeah, so this is just the fundamentals. A DNA duplex, which is like a single 2-base pair strand, combine together, make DNA tiles, and these tiles self-assemble into DNA structures. So yeah. And thermodynamics, where does temperature come into all this? So temperature actually affects the nucleic acid structure of these double helical strands. And so under certain temporal conditions, researchers can manipulate the speed at which these structures self-assemble, making the process much, much more efficient. Going from 12 hours to self-assemble to 10 minutes. So they're saving lots and lots of time by studying this. So this is where I spend most of my time, the work before preparing the samples. So to purify a DNA strand, what you gotta do is you gotta take the impure strand solution that IDT sells us, and then we vortex the strand to ensure that the solution is homogeneous. Homogeneous means it's all under one solution, there's not extra particles. And then we add water to the strand, and then we centrifuge it to prepare for use. And the nanodrop machine is a machine I use to, so another thing is, a lot of the strands we bought, we didn't even use. We threw away right away because they weren't concentrated enough. Because researchers mess up a lot, like there's a lot of mistakes in the lab actually. So many of the strands that we used were actually thrown away. We chose the best strand based on this machine. We put the solution under the nanodrop and we measured the concentration, and the molecule with the highest concentration was the one that we used. So lots of money. Loading die. So in order to, the solution is clear, so it's very hard to study. So so we can see it, we add a loading die to the solution, which makes the color green so we can actually see it. Because this solution is clear and so is the gel we make to hold it in. So we wouldn't be able to see it until we had this loading die. So this is another important part of the DNA. And so separating strands for study. This is where I excel at making gels. I think I'm better at making gels than the actual professors now. So we use the denaturing page gel, which is this one, for larger strands, which is mainly what we did in the thermodynamic field. And then we use agarose gel, which is right there, for smaller strands. So those are just the two different types of gels that we use. So these are the components of the gel. TEMED, I'm not even going to try to say that. APS, ammonium parasulfate, 0% polyacrylamide urea buffer solution, which is a mixture of a mixture of urea, 10x TDE buffer, and diluted water. And then a 20% polyacrylamide solution. So more about TEMED, a lot of bad experiences with this. It's a neurotoxin, it's corrosive, and it's highly flammable. But it's essential to the gel because it enhances the polymerization of it, along with APS. And 10% APS, which is ammonium parasulfate, it triggers the polymerization of the gel, and it needs to be frozen at negative 20 degrees Celsius at all times, and then unfrozen just before use, or else the structure will mess up. Urea buffer and 20% acrylamide solution. These are the two. This is the base of the gel. So this is the majority of the gel. The TEMED and the APS polymerize this gel, but the majority of the actual gel itself is these two, the urea buffer and 20% acrylamide solution. And gel electrophoresis is the process by which we use pretty much electricity to separate the macromolecules, which is in this case DNA, to study. So it isolates the needed molecule from the other components of the gel. And long DNA strands are studied in page gel, which that's repetitive. qPCR. This is the machine that we actually use to measure the thermodynamics of these DNA strands, because it monitors the changing fluorescence. And if you guys remember the loading die that we used in the solutions, under this machine, based on how much we increase the temperature and how the fluorescence changes, we can measure the thermodynamic properties of the DNA itself. So this is just the machine we use. I never actually got to use it, I just got to watch them use it. But it's pretty cool stuff. And the fruits of our labor. So researchers at ASU were actually able to create structures like these more efficiently and effectively, with way more than before. This is the smiley face that shows you. That's a boss for, there's I don't know, sometimes they create structures just for fun. And that's interesting about research is that I thought it was going to be really serious and oh, there's deadlines we have to meet. And there is deadlines, but these researchers know how to have fun a little bit with their work. So that would really impress me and kind of made me like research a little bit more. So yeah, these are just some of the structures that were made using DNA thermodynamics. And so yeah, it's still an emerging field. Like, don't get me wrong, there's not very many like practical uses yet, other than abraxane, which is the breast cancer drug. But researchers have learned vast amounts of how well DNA structures like aggregate and denature under certain conditions. So we've learned more and more about the properties of the molecule just at ASU itself in the past 4 years. So where do we go from here? So although researchers at the Biodesign Institute have made significant advances, there's still a lot more to be learned. But the future is promising because the research only started 4 years ago, and they've already synthesized a cancer drug. So research is definitely promising. So acknowledgements, I'd just like to thank Professors Yan Liu and Hao Yang for giving me the opportunity to conduct research, and my mentor Shou Jing for being there for me, and my school mentor Ms. Raj for helping me throughout the process, and of course my SRP coordinator Mr. Valley for landing me this placement. And these are my reference. Questions?
What was the toughest part of your SRP?
Toughest part of my SRP was getting over the fact that I didn't want to do it originally, but then I got over the idea and I just decided to go through with it. So, and that was pretty rewarding because like it was bad until I just gave up on trying to not do it and focus more on what can I get out of this instead of why am I doing this. So that was definitely the hardest part, getting over that barrier of like I don't know what I'm doing. Sorry.
So even though you were just making the gels, knowing what you know, do you feel it was worth your time and effort?
I would say definitely if I was going to major in biochemistry or any field of biology, and I am sort of, I'm doing biomedical engineering actually, so overall it was rewarding just for the experience of research itself. I mean, like even forgetting all of like the complex things that are going on here, just the experience of research is an extremely valuable one and one that I believe I needed to know. Because if I joined research in college or after college without knowing how it was like and I didn't like it, it's not good, right? So like I don't know, the experience for me was like more than like what I actually learned. Because to be honest, I didn't learn a whole lot because it's pretty complex stuff. But I learned, I learned, but I didn't learn about the actual like process itself. I learned more about like research and like how to like carry yourself in a research lab and stuff like that. Mr. Rob?
What would you suggest, what advice would you give, let me put it that way, what advice would you give to students who might indicate an interest in wanting to work at the Biodesign Institute, something similar next year? How would you like to have been better prepared going in?
Definitely don't take research for granted. You're not just going to go in there and know everything. That's the biggest misconception I made. I thought it was going to be pretty easy to learn this. I was dead wrong. And do your reading beforehand. Definitely research beforehand. You want to be prepared when you go in the lab. You don't want to go in not knowing what you're doing because research is very efficient and it's time consuming. So you want to know what you're doing so you can get the most out of your time there.
I have two questions. I'll start with the first one. I'm absolutely fascinated by the idea of solar panels using DNA instead of silicon cells. And I know you said there weren't any, you were expecting the big discoveries every day. Did you get to see any research moving in that direction?
Actually, my whole time there, they were more focused on learning about the actual properties of the DNA and stuff rather than moving towards these goals because these, I wouldn't say they're far-fetched, but these subordinate goals are very far down the timeline, because it's like the majority of research conducted there is like very they take it one step at a time, and they don't really look forward to all these breakthroughs. They're just trying to figure out for the next day what they're going to do.
So not something I can expect to have for Christmas.
I wouldn't count it. If it does, I'll let you know.
Okay. My second question, when you're working with chemicals that are neurotoxin, highly flammable, there's all kinds of, for me, interest in any mishaps. What did you have or see any mishaps with any of these things in the lab? And can you give us a brief?
Yeah, actually, my good friend AP over there got some on his eyebrow, and it burned someone's eyebrow. So, yeah, there have been mishaps, but we've learned from that, right, AP? So, yeah, but other than that, like, no serious mishaps. I mean, just
It's not serious. How patient were the scientists as far as working with you in the lab?
Oh, they were extremely patient. Like, if I was them, I would not put up with me. They were extremely helpful. They were so helpful in this process. If I didn't have my mentors, my on-site mentors, and the professors helping me out every day, I would be lost. Because the stuff they're doing, I had no clue about that even existed. But they were so patient, and they took their time out of their day to explain it to us. And they're pretty busy people because they're doing groundbreaking research. So, yeah, they were very, very helpful. Mr. Rock?
So I know that you went into this and it was a little overwhelming at first. What was the one thing that got you to keep going and finish the project?
Well, okay, so I think in the middle of March, I had a meeting with Mr. Valley and my mentor, and AP's mentor and AP. And one thing that really hit me was like, I felt like I was useless kind of in the lab because I didn't know anything, but then I realized like what Mr. Valley was saying, like the experience and like the knowledge is half of it. And now I know what research is, so like even if I didn't enjoy my experience, which I did towards the end, I would know not to do research. He said it was a good learning experience, which is what truly led me to finish this project, because even if I didn't feel that sense of self-fulfillment, I still got the experience needed to realize what research is. Ms. Ross?
I understand that your project is ongoing, but you talked about so much about the DNA. What's the source of the DNA?
Oh, we get it from IDT. Which is, oh, sorry, let me explain that. IDT is the company where we buy the DNA strands from, and I'm actually not entirely sure where they get their DNA from, but they actually supply us with custom nucleic acids, so it's not even like DNA at its core, it's like modified DNA. So that's where we get our DNA from, but I'm not sure where they synthesize their DNA from.
So you purchased it.
Yeah, we purchased it.
And you talked about nano, the machine.
The nanodrop machine?
What's the purpose of that?
Oh, it measures the concentration of the DNA solution, so we know which strands are the most efficient to study, the ones with the highest concentration. So ones with lower concentration than the colloid we don't use because it's a waste of time. It's not going to produce good results.
So how do you see the outcome of that machine, the result of that machine?
Oh, okay. So it's linked to a computer, right? And then once you put the solution on the balance, you close the machine, and you click measure on the computer. The computer comes up with results about the concentration. Mr. Ron?
Yeah, would you say, so I get the feeling from your presentation that they're going into it trying to understand the fundamental aspects of DNA and thermonuclear or thermodynamics and everything. Would you say that many of the practical applications that will come in the future would be discovered accidentally, or would it be because the one with breast cancer drug delivery did they start up with that end goal in mind? Or how?
Actually, the majority of to answer your question, the majority of discoveries are found accidentally, but a lot of them stem from what researchers already know about the structure itself. So once they find like something that's not going as they expected, they can use their fundamentals that they learned from studying the thermodynamics of DNA to kind of link those two together, and that leads towards discovery. Dr. Majeski?
The smiley face and the face, how big are
They're actually extremely small. They're 9 nanometers length, and the smiley face costed $1,200, like I said, and then the vase costed, I think, a lot more because it was bigger. So, yeah, they're really, really small. Like, you can't see them until you use it at AFM or another like machine to measure a microscope. Yeah, they're very small.
One more, last question.
On that same topic, smiley face, do you think they were trying to figure out whether they can manipulate them into certain shapes, or was there some purpose behind that?
Yeah, that's actually what it is. Those smiley faces, the researchers let the DNA self-assemble under conditions that they set. Those smiley faces assemble themselves under certain conditions set by the researchers. The thermodynamic properties of the DNA allowed them to self-assemble under certain temperatures. So yeah, you can manipulate DNA to make structures that you want.